All right, let's dive into one of the most incredible, most mind-boggling puzzles in all of biology. We're talking about protein folding. You know, it's something that's happening inside your body, literally trillions of times a second. But it's so ridiculously complex that it's still one of science's biggest mysteries. So, today we're going to try to unravel it.
So here's the question, the big one that keeps scientists up at night. You start with this long, kind of floppy chain of molecules, and then in less than a blink of an eye, it folds itself into a perfect, super-specific 3D machine that can do everything from digest your lunch to fight off a virus. How on earth does it know how to do that every single time without a mistake?
Just look at the difference here. It's pretty stark. On the one hand, you have what's basically just a string of beads, what scientists call a random coil. It's useless, it's formless, and on the other hand, you get this intricate, functional piece of machinery. The jump from A to B is just a masterpiece of nature's engineering. So let's figure out how it's built.
To solve any puzzle, right, you have to start with the pieces. And for proteins, those fundamental pieces are called amino acids. These little guys are the building blocks, and as we're about to see, they hold all the secrets to this amazing act of self-assembly.
So, every single amino acid has the same basic skeleton, you could call it, a central carbon, an amino group, and a carboxylic group. But each one has a special part that makes it unique, and that's the side chain, which we just call the R group. This little R group is everything. It's what gives each amino acid its own chemical personality.
So how many of these unique building blocks does life need to create pretty much every protein you can imagine? Just 20. That's it. It's like a chemical alphabet with only 20 letters, and with those letters, nature writes the story for this unbelievable variety of protein machines. Okay, so these amino acids don't just hang out by themselves. They get linked together by this super strong connection called a peptide bond. This bond creates the backbone of the protein, stringing all those amino acids together one after another into a long strand we call a polypeptide. And there it is, our beaded necklace.
So, we have the chain. But how does it know how to fold? Well, this is where those R groups, those chemical personalities, really come into play. See, it's not just a random string of beads. It's more like a cast of characters in a play, and each one has a role to fill and a specific place they need to be in the final structure.
You can basically sort these characters by one simple preference, how they feel about water. You've got the ones that are charged or polar. They're the social butterflies. They love water. So you'll usually find them on the outside of the protein, just hanging out with the watery environment inside our cells. But then you have the nonpolar ones, the hydrophobic ones. They hate water. They're the wallflowers, and their first instinct is to run away from it and bury themselves deep inside the protein's core. And that simple push and pull is one of the biggest forces driving the whole folding process.
Now, besides that pushing and pulling, there are two kinds of really strong, permanent bonds that help lock the final shape in place. We already met the peptide bond. That's what makes the chain itself. But there's another super important one, the disulfide linkage. The best way to think of it is like a powerful staple that can form between two very specific amino acids, locking parts of the chain together, sometimes from really far apart.
This all sounds pretty logical, but how do we actually know, for sure, that the sequence of amino acids is what dictates the final shape? Well, the answer comes from this absolutely brilliant and honestly astonishing experiment that just laid the secret bare for everyone to see.
So what the scientists did was they took this one specific, fully working protein. It was made up of 124 amino acids, and, really important, its shape was held together by 4 very specific disulfide bonds, those structural staples we were just talking about. It was in its perfect, active, native state.
The experiment itself was so simple, but the result was profound. First, they added some chemicals to completely wreck the protein's shape. They broke those disulfide staples, they unraveled the whole thing, and turned it into that useless, floppy, random coil. Then, and this is the incredible part, they just took the chemicals away. They did nothing else. And what happened? The protein, all by itself, folded right back into its exact original shape, reformed the 4 correct staples, and started working again perfectly.
This was the smoking gun. This experiment proved it. Everything the protein needs to know to build its final, perfect 3D machine is already written into that simple, one-dimensional line of amino acids. The blueprint for the complex machine is encoded right there in the simple string of letters. There's no external foreman, no instruction manual. The chain is the instructions.
And that just brilliantly solves one part of the puzzle.
We know what holds the information. But in a way, it makes the mystery of how it all actually happens so fast and so perfectly even deeper.
This is a direct quote from the source material, and it's just this powerful, humbling reminder of how much we still have left to figure out. Even though we know this fundamental principle that the sequence is the code, the actual path a protein takes from a straight line to its final shape is so complex, it's still considered one of the great unsolved problems in science.
And that really just leaves us with this final thought to chew on. We have the alphabet, we have the words, we know the 1D sequence contains all the instructions for this 3D masterpiece. But how does nature actually read those instructions and perform this incredible act of chemical origami flawlessly and in the blink of an eye, trillions upon trillions of times? Well, that is a puzzle for the next generation of scientists to solve. Thanks for tuning in.